Alright guys, so this is my presentation. I have 5 slides prepared, but I just left the title slide in there so you guys could just be reminded of what I'm presenting on. So the effects of urea and tyrosine kinase activity on epithelial cells. So there we go. So the first slide I'd like to present on is the slide following my introductory and my background slides. So this is my workflow slide, which shows what I've been doing in the lab from the very beginning. So everyone kind of started with the sample collection, and after that the cells that we collected from the samples were cultured, and then a urea assay was done by me on the raw cells, just kind of like straight out of being pelleted and centrifuged and all that kinds of stuff. And then results were obtained from that assay. And then following that, we then exposed the cells that had the highest cell counts to e-cigarette liquid with various amounts of nicotine in them. And once that exposure was done, another urea assay was conducted on those cells. And then in the future, we'd hope to be doing a tyrosine kinase activity on those same cells, but those results haven't been produced yet. So right now I'm going to talk about the results that have been obtained so far with the urea assay. So these are just some methods used to obtain the cell samples, So we had 9 participants total, and each participant produced 2 washes or 2 samples, really. So prior to both samples, the person's mouth was washed with approximately 20 mils of mouthwash, and then 2 mouthwashes were done relatively the same way. The cells were obtained after brushing your teeth with a sterile toothbrush like you would any other day, and then rinsing your mouth with purified water, then spitting that into a small container, and then rinsing the toothbrush with additional purified water, and whatever is collected from that is also put into the small container. And then after a 30-minute time period, the exact same thing was done, except the cells were collected in another, like a different container. So in total, we had about 18 cell samples labeled 1A, 1B, 2A, 2B, and the A and B's just represented the first and second washes. And from there, we then pelleted the cells that we collected from each of the samples using the centrifuge, and then they were rinsed 3 times with phosphate buffered saline or PBS solution, and then either cultured immediately or they were stored in the -80 freezer until we were ready to culture them. So after all this had been done and the cells had been cultured, like I said, we then ran the urea assay on them. And these are the results from the A samples, which is the very first wash that each of the participants did. So we have 1A through 9A, and the red and yellow colors represent the different optical density, like readings we did. And it's initially supposed to be done at 430, but our spec only runs at either 405 or 450. So these represent the 2 varying like levels that they were read at using the spec. And as you can see from this data, the 4A actually ended up having the highest urea content, which was really interesting. And there were a few others close behind, but that's just to show you what the raw samples without being exposed to any liquid showed us. And then these samples or results were from the samples of the second washer, the B samples. And it's also interesting to observe here because as opposed to the 4B being really high in the urea content, we actually see the 8B and the 9B being pretty high when they weren't as high in the first wash. So this is something interesting to know, and again, these weren't exposed to any like nicotine or liquid. These were just some preliminary results that we did to get a feel of what the results could produce, and so we could be more comfortable with the collection and the process when we did the assays on the hard, like exposed cell samples. So what we did before doing this next assay was we took the cell sample with the highest cell count or the highest cell content, which ended up being the sample 5B. So that was the second wash from participant number 5, and we exposed them to 4 different e-cig or vaping liquids with different nicotine contents. The first was 0 mg, the next was 3 mg, and then 6 and 25 mg. And then we also had a control which was exposed to no liquid at all, so we could have just like a baseline of what we wanted to see. And so from these results, we can see that they're all pretty similar, but the 0 mg seems to have one of the higher urea contents. And in the future, we're going to try and do more exposures for this 5B sample, and then we're also going to be running exposures and urea assays on the 1A sample, which was the next highest in cell counts. So those are basically the slides I have so far. I just wanted a little bit of feedback on the flow and maybe if there was some data that was more pertinent to show than other data, but those are the results slides and the results kind of portion that I have right now. So that is it. Thank you.